Hi everyone, my name is Nur Shuhadah binti Muhammad Razwan, and today I will be presenting the introduction for our experiment titled Separation of Plant Pigments Through Paper Chromatography. First, I will explain the background of this experiment. Chromatography is an important analytical technique used in chemistry and biology to separate, identify, and analyze the components of a mixture. In plants, leaves contain a variety of pigments that play an essential role in photosynthesis, which is the process by which plants produce their own food. Although most leaves appear green, they actually contain several pigments with different colors and functions, such as chlorophylls and carotenoids. These pigments differ in their chemical structure and purity, which affects how they interact with solvents with solid surface. By using chromatography, these pigments can be separated and observed clearly, allowing us to understand that plant leaves are made up of more than just one pigment. Next, I will describe the technique used in this experiment, which is paper chromatography. Paper chromatography is a simple and widely used method for separating colored substance. In this technique, a small spot of plant pigment extract is applied near the bottom of a strip of chromatography paper. The paper is then placed vertically near the bottom of a strip of chromatography paper. The paper is then placed vertically in a container with a suitable solvent known as the mobile phase, ensuring that the pigment spot does not touch the solvent directly. As the solvent moves upward through the paper by capillary action, it carries the pigments along with it. The chromatography paper acts as the stationary phase. Different pigments travel at different rates depending on their solubility in the solvent and their attraction to the paper. As a result, the pigments separate into distinct bands at different heights on the paper. Now, I will explain the plant pigments observed and their colors. One of the main pigments observed is chlorophyll A, which appears blue-green in color. Chlorophyll A is the primary photosynthetic pigment found in plants. It absorbs light energy mainly from the blue and red region of the light spectrum and plays a major role in converting light energy into chemical energy during photosynthesis. Another pigment observed is chlorophyll B, which appears yellow-green in color. Chlorophyll B is known as an accessory pigment. Its function is to absorb light energy that chlorophyll A cannot absorb efficiently and transfer this energy to chlorophyll A. This process helps to increase the overall efficiency of photosynthesis.
Next, the objective of these experiments are to separate and identify different plant pigments using paper chromatography and to determine the relative affinity of each pigment towards the mobile phase and the stationary phase. Chromatography is a technique used to separate a mixture by distributing its components between two phases, namely the stationary phase and the mobile phase. The stationary phase acts as a constraint that slows down the movement of certain components in the mixture based on their interaction with the phase. Meanwhile, the mobile phase carries the components of the mixture through the medium being used. The retention factor or RF value is defined as the ratio of the distance traveled by a solute to the distance traveled by the solvent front.
For the methodology, this experiment was carried out using paper chromatography to separate plant pigments. This procedure involved four main steps. First, preparing the plant extract. Second, preparing the chromatography paper. Third, developing the chromatogram. And finally, observing and recording the results. First, fresh green leaves were washed to remove dirt and then dried. The leaves were ground using a mortar and pestle to break down the plant cells. A small amount of organic solvent such as acetone or ethanol was added to dissolve the pigments. The mixture was then filtered to obtain a clear plant pigment extract. Next, a strip of chromatography paper was prepared. A straight starting line was drawn using a pencil about 1 to 2 cm from the bottom of the paper. Pencil was used instead of pen because ink may dissolve in the solvent. The plant extract was then carefully spotted onto the line using a capillary tube and allowed to dry. After that, a small amount of solvent was poured into a chromatography chamber. The chromatography paper was placed inside the chamber with the solvent level below the starting line. The chamber was covered to prevent solvent evaporation. The solvent was allowed to move up the paper by capillary action, carrying the pigments along with it. Once the solvent had nearly reached the top of the paper, the chromatogram was removed. The solvent front was marked immediately. Different color pigment bands were observed at different heights. The colors and distance traveled by each pigment were recorded for further analysis. In conclusion, this method successfully separated the plant pigments using paper chromatography. The separated pigment bands were then used for further discussion and analysis in this experiment.
Based on the methodology discussed earlier, all of these example chromatograms illustrate the outcome of the paper chromatography process used to separate plant pigments. In each case, the plant extract was initially applied at the origin line near the bottom of the paper. As the chromatography paper was placed in the solvent traveled upward by capillary action, carrying the pigments along with it. Across all of these chromatograms, a consistent separation pattern can be observed. 4 distinct pigment bands are visible at different heights on the paper, corresponding to keratin, centerfill, chlorophyll A, and chlorophyll B. The solvent front is clearly indicated at the top, showing the maximum distance traveled by the mobile phase. In all examples, keratin is observed closest to the solvent front, indicating that it traveled the furthest distance and is the least polar pigment. Centerfill migrated slightly less, followed by chlorophyll A, while chlorophyll B remained closest to the origin line due to its higher polarity and stronger interaction with the stationary phase. The similarity in pigment order and separation across all chromatograms demonstrates that paper chromatography provides reliable and reproducible results. This observed migration distance from the basis for calculating RF values, which will be discussed in the next slide. Based on the reported data, this slide summarizes the results of plant pigment separation together with the calculated RF values. The table shows 4 plant pigments, which are keratin, centerfill, chlorophyll A, and chlorophyll B, along with their respective color, migration distance, and RF values. The distance traveled by each pigment was measured from the origin line to the center of each pigment band, while the solvent front traveled a constant distance of 63 ml. Keratin traveled the furthest distance at 59 ml, followed by centerfill at 56 ml. Chlorophyll A and chlorophyll B traveled shorter distance of 29 ml and 14 ml respectively. Using these values, the RF value of each pigment was calculated using the equation, which is the distance traveled by the solute divided by the distance traveled by the solvent. For example, the RF value of keratin was obtained by dividing 59 ml by 63 ml, giving an RF value of 0.94. The same method was applied to centerfill, chlorophyll A, and chlorophyll B to obtain their respective RF values. From the results, keratin recorded the highest RF value, followed by centerfill, chlorophyll A, and chlorophyll B. This trend indicates that keratin is the least polar pigment and has the strongest affinity towards the mobile phase, allowing it to migrate further up the chromatography paper. In contrast, chlorophyll B has the lowest RF value, indicating stronger interaction with the stationary phase and higher polarity. Overall, the differences in RF values provides quantitative evidence that plant pigments can be effectively separated and distinguished using paper chromatography based on their polarity and interactions with the stationary and mobile phases. To conclude, this experiment clearly demonstrates the principle of paper chromatography in separating plant pigments. Paper chromatography operates based on the differential distribution of compounds between a stationary phase, which is the chromatography paper, and a mobile phase, which is the solvent. As the solvent moves up the paper through capillary action, the plant pigments are carried along at different rates. However, not all pigments migrate the same distance. This is because each pigment has a different polarity and affinity towards the stationary and mobile phases. Less polar pigments, such as carotenoids, interact more strongly with the mobile phase and therefore travel further up the paper.
In contrast, more polar pigments, such as chlorophyll B, interact more strongly with the paper and move a shorter distance. This difference in migration results in the formation of distinct colored bands on the chromatography paper. The separation observed confirms that plant leaves contain multiple pigments rather than a single coloring compound. Additionally, the calculated RF values further support this observation, as each pigment exhibits a unique RF value that reflects its chemical behavior during the chromatographic process. Overall, this experiment demonstrates that paper chromatography is a simple yet effective analytical technique for separating and analyzing plant pigments based on their polarity and interactions with the stationary and mobile phases.